feeding center. This feeding center stimulates our appetite to take up food. When we take up food, we increase our fat stores. That expands the radius of the somewhat spherical adipocytes or fat cells in the body. And when their cell membranes get stretched by increased triglyceride stores in globules in the adipocytes, they secrete leptin, probably through exocytosis due to stretch-activated channels that activate voltage-gated calcium channels and stimulate exocytosis. So leptin goes up to these neurons in the central nervous system, inhibiting their neuropeptide Y release, whereas ghrelin from an empty stomach is one of the other neuropeptides and hormones, and there happen to be many, that excite positive sign neuropeptide release into the hypothalamic feeding center. Others that also stimulate these, I don't have on hand. So you might say our calories in is our energy in, and it's our diet, and that's potential energy coming in the form of chemical bonds that can be broken to give off energy. Typically, if they're exothermic, they give off heat. And that dietary input, if it isn't balanced by work, by transport across the membranes, muscle contraction, and metabolism, as well as heat released from the body from evaporation of sweat and conduction and some infrared emission, unregulated heat loss. If those two don't balance, then our body is going to either go up or down in body mass. That's the first article of faith for dietitians. There's probably fine tuning to that because physiology that isn't really well understood or published yet, say for instance, regulation of feces production through gastrointestinal mechanisms, and also the biome, the bacteria living in our intestines that would affect how calories coming in end up getting utilized. Some of those fine tunings we'll learn about in the future, but not in this course. Question number 4. How many calories are used per hour for each kilogram for a man at rest? How about a woman? Why is there a difference? If 1 calorie of food is burned in a calorimeter, how much will it warm up 1 liter of water? If you can measure someone's VO2, their oxygen volume consumption rate, while they are exercising, what do you learn about their metabolic rate?
Hey, Dr. B, did you tell them about the video for April 3rd? And I missed that. I might need to just announce that.
No.
Okay, let me go ahead. So you guys, I accidentally combined this lecture and the metabolism lecture into one video. So you do not have a pre-lecture video and you need to watch for Friday. I just sent out an email for you guys, so hopefully you get that too. But yeah, so just go ahead. The quiz is open for Friday. You can just go ahead and take that whenever you want, saying you did it because you did. I just screwed up and combined the 2 lectures into one. So there'll be a video for Monday again, but nothing until Monday. So you're good.
Thanks, Morgan. I forgot space that. It was my fault that Morgan did that because I told him that we only had 1 lecture on this PowerPoint. So he did the whole PowerPoint into 1 lecture. But that means that all of you have already put a 1 in the exam for today can go ahead and put a 1 in the exam for Friday because you already saw Friday's video. While we're off the subject of this question, we will plan to allow you to take the final exam through the exams tab in our Learning Suite course. anytime during the final exam week between Friday and I believe it's Wednesday or Thursday, whatever the final day of finals is. I promised you we'd announce that, and we'll post it as well. Okay, Joseph says that at rest, men will use 1 calorie per hour per kilogram. Wait a minute. This says kcal. Why? Because the chemist's kcal is the dietitian's cal with a capital C. So 1 kcal is the same as 1 cal with a capital C. Every hour for every kilogram. So the 70 kilogram man will have a basal metabolic rate, a BMR, of 70 calories per hour, and there's 24 hours in a day. So 7 times 24, 141, I think it's 1,680, if I'm not mistaken, calories per day would be the basal metabolic rate for a 70 kilogram man, each kilogram weighing 2.2 pounds. That'd be a 154-pound man. Females, less, and that's because they have a bigger percentage of fat relative to muscle, and fat doesn't burn calories as much as muscle does. There's just this kind of basal turnover of ATP in skeletal muscle that's constantly getting used up, which isn't needed to keep our fat stores constant. Hormones also affect the difference. If you burn up 1 calorie of food equivalent that would have 1 calorie in it, in a calorimeter, it'll heat up a liter of water 1 degree Celsius. That's what 1C means. 1 degree Celsius or centigrade. So that's what we mean by a calorie. That's the potential energy stored in the bonds. You burn them up, turn them into oxidized molecules, and release the heat. It's a little bit more complicated than that from the chemist's point of view, but we'll leave it at that for our point of view. If you could measure somebody's oxygen consumption, VO2, you would be detecting how many calories they're using, because that's a good approximation. There's 4.5 to 5 calories used up for every liter of oxygen that is burned. Now, it can be done, and exercise physiologists do it all the time. We're going to come back to this quite a bit. We're going to talk about the VO2 max, they call it, the maximum oxygen consumption rate. How much could your mitochondria burn if they were getting the maximum amount of oxygen in? But you have to not only know what fraction of the air is oxygen, what's the tidal volume, how big is the tidal volume when you're exercising. So it's more than half a liter when you're exercising. It could be as much as the vital capacity when you're exercising full out. But not only that, you have to know how much oxygen comes out in each breath, because not all that comes in comes out, and that's the trickier part of the equation. Nevertheless, once you measure it, it can be done. You can get the VO2 max, and it can max out at 3.5 liters per minute in a well-trained male, or 2 liters per minute in a well-trained female. I shouldn't have said well-trained, I should have said moderately well-trained, because with extensive training, you can double that. The respiratory quotient is the production of CO2 divided by the consumption of oxygen. How many oxygen molecules, O2 molecules, end up attached to a carbon and coming out again? It's kind of interesting, not to, that carbohydrates such as starches and glycogen end up giving 1 CO2 for every oxygen, whereas proteins, they're not as efficient. They give 0.8 and fats 0.7. We use fat and glycogen to store potential energy in the form of chemical bonds in the body, which was a separate issue not in question. Next question, question 5. What words are used to describe triglyceride buildup, triglyceride breakdown, glycogen buildup, glycogen breakdown, and synthesis of glucose from amino acids and breakdown of glucose? Go. Welcome, Derek. Craig, glad you're here. Crowd's building up a little bit here, Morgan. Okay. Getting some latecomers. Glad you could make it. Answer is triglyceride buildup is lipogenesis. Genesis of lipid. Lipogenesis involves free fatty acids combining with glycerol to make triglycerides, which are our fat stores. The free fatty acid pool in this Rube Goldberg machine type of diagram is referring to the free fatty acid concentration in the plasma of the bloodstream. Glucose concentration in the plasma is called the glucose pool. The amino acid concentration is called the amino acid pool. And these concentrations in our bloodstream feed our whole body, our brain, the metabolism of our tissues, and they come down and then get turned into body proteins, and we eat carbohydrates. They get broken down. I hope you can all hear me. My unstable internet sign just came on. The question of why do we break them down and then build them back up again? Doesn't that seem a little inefficient? It's a reasonable question, but the simple answer is that we can take in things that are made from other organisms and rebuild them into our organism type of proteins so that we can be immune to the other organisms in our blood, for instance, or in defenses throughout our body, but build our own kind of protein. Now, that's not as much the case for fats or for the storage of glucose in the form of starches in plants and glycogen in animals, but it's, here I should point over here, but there are other ways, other features that make this worth doing for both fats and glucose as well, which we'll come back to in the future. What about glycogen buildup and breakdown? We've got glycogenesis, building up our glycogen stores, and glycogenolysis, breaking down our glycogen stores. Synthesis of glucose from amino acids. We've got gluconeogenesis, taking certain amino acids and adding glucose to the pool after deaminating them. And breakdown of glucose, we have glucose being broken down through glycolysis in most tissues. So with that introduction now, let's focus a bit on glycolysis first. Glycogenolysis produces glucose because glycogen is a polymer of glucose, like starch is a polymer of sugars. Glycogenolysis produces glucose, and that can be phosphorylated for glycolysis in most tissues. The liver behaves a little differently. We'll come back more to that next lecture. But it still, this is still true for the liver too. The phosphate is moved to ADP, and the hexatriol is broken down to 2 3-carbon pyruvates. So by hexatriol, I'm talking about a 6-hydroxide, 6-carbon chain that is usually linked into a cyclohexane structure to make something like glucose. the way the hydroxides stick off determine whether it's glucose, maltose, or not maltose, glucose, lactose, no, fructose, glucose, fructose, or galactose. The 3-carbon pyruvates can be converted to lactate, which is also 3 carbons, just has an extra hydrogen added to one of the oxygens in anaerobic conditions. So question number 1, how does pyruvate get into the mitochondrion to acetylate coenzyme A? The acetate then being contributed to the citric acid cycle and giving off the CO2. Question was, how does it get in? Question number 2 in this question is, what is the word for fatty acid breakdown in the mitochondrion? And I'm not talking about lipolysis here, which is triglyceride breakdown in the adipocyte. I'm talking about fatty acid breakdown in the mitochondrion of the metabolizing tissue cells. Number 3, what is the word for amino acid deamination that's used to produce pyruvate, which can then pass backwards through the glycolysis chain to produce glucose and have it be sent out into the bloodstream from the cytoplasm. Go. Answer is, thanks to Josh Ballard, by the way, for resupplying this word for me that I didn't remember. I trust you, Josh. Pyruvate translocase transfers pyruvate, an acid without an amine group, through the mitochondrial membrane to get into the mitochondria where it can acetate coenzyme A. Acetyl-CoA can be made from fatty acids as well when they're broken down by beta oxidation. In the process, some alkyl ketones are produced, and that's what produces ketosis, which the ketosis diet fans are enthusiastic about. They want to use up your fatty acids, make more ketone bodies, and as much as possible, be using those for energy in the brain and elsewhere. The breakdown of these lipids is called beta oxidation. The breakdown of amino acids, deaminating them to make acids, which eventually can be made into pyruvate, a very short 3-carbon acid, with a ketone and a carboxylate, which can then be passed backwards through the glycolysis enzymatic pathway to make glucose 6-phosphate, which in the liver can be converted to glucose and secreted into the bloodstream to keep our glucose concentrations up when they get low. So this is called gluconeogenesis, the production of glucose de novo from amino acids. And it's one mechanism that the body uses when it needs to break down our proteins and make glucose to keep our brain alive. We don't use this until last resort, generally. We reserve our proteins as long as we can. Question number 7. Fat is stored in adipocytes as globules of triglyceride that essentially fill the interior of the cell. It's like they're the cytoplasm, but you can't think of fat as cytoplasm. The cytoplasm is electrolyte solution that would be surrounding the fat globule. In lipolysis, these globules of triglyceride are broken down into a monoglyceride and 2 fatty acid molecules, which can easily permeate membranes to get into the blood plasma. Don't forget, too, that we learned last time that chylomicrons from the chyle, which were exported to the basolateral membrane of the epithelial cells lining the intestine, which is how the epithelial cells took bile emulsified fat and turned them into small globules of fat that could be transported through the lymph into the bloodstream and eventually passed to the liver and other tissues. These chylomicrons are broken down by a protein in the membrane of the endothelial cells lining blood vessels called lipoprotein lipase. How are the fats, including cholesterol from the chylomicrons, transported in the blood? What cells process HDLs? How about LDLs? What proteins, and I might say synthesized by the liver, bind up lipids, including fatty acids, monoglycerides, and cholesterol, to form high-density lipoproteins, HDLs, and low-density lipoproteins, LDLs. I'm assuming that you're familiar with these terms, HDL and LDL, from somewhere else, from medicine, because they're very important for physicians who care about atherosclerosis, and we did mention them previously in that context. It seems that high HDL protects us from atherosclerosis, whereas high LDL causes atherosclerosis. So that's why we're taking a more focused look at HDLs and LDLs in the next slide. Everybody in? Welcome, G3. Glad you could come.
Dr. Busaid, I don't see your screen being shared anymore. It just says energy utilization. Is the PowerPoint still up?
The PowerPoint is still on, but it says my screen sharing is paused. Let me see how I unpause it.
Yeah, I've just seen the same slide for the whole lecture.
I am so sorry for that. Let's see. How do I unpause screen sharing, you ask? Recording on this computer. There's a stop share button. Oh, here's a resume share button.
Yay.
How's that look?
That's a lot better.
Oh, thank you. Shall we go back and start again? No.
Maybe, quickly.
Evidently, either all of you have these things so well in your minds from Morgan's lecture that you don't need to see the PowerPoint, or you've got your PowerPoint open on the side and you don't even look at the one I'm projecting. But for those who have neither of those to be the case, heat balance is radiation in, conduction out, convection out, radiation out, evaporation out, and internal heat production in. Regulatory center has the sympathetic cholinergic neurons and sympathetic adrenergic neurons that we are responsible differentially for responding to cold, conserving our heat, or hot, getting rid of heat. Energy balance is the balance of dietary calories in, potential energy from calories, and heat and work out. Our hypothalamic feeding center does the fine tuning to keep our body mass approximately constant. We have basal metabolic rate. We know what calories are by this definition, and our body consumes oxygen at that rate. This Rube Goldberg machine describes the amino acid pool, the glucose pool, and the free fatty acid pool, I should have pointed here, in the bloodstream, how those feed into making up metabolism in our tissues and our brain, making up new proteins, making up lipids in our adipocytes. We talked about glycolysis here and the role of pyruvate driving the fatty acid cycle there. We kind of skipped. I'm glad we went back to this. Here's the CO2 being produced. So our CO2 production rate, VCO2, is coming from this. We also have from the citric acid cycle, the electron transport chain stimulated by high energy electrons and protons that use up our oxygen. Here's our VO2 being used up in the process of making ATP. We are currently talking about this slide. Chylomicrons are broken down by lipoprotein lipase, lipoprotein lipase. They are little particles that have proteinaceous remnants that can get processed in liver cells. The free fatty acids and monoglycerides, the cholesterol that comes with them ends up in the blood and being processed. The free fatty acids go to the adipocytes where they can be turned into triglycerides for storage, and when needed, broken down to make the free fatty acids among monoglycerides again that can be used in beta oxidation through the mitochondria for energy. Cholesterol being used to synthesize things like aldosterone, testosterone, progesterone, those kinds of sterone hormones. But the key to this question is there's a focus here on HDL and LDL. High density lipoproteins are made from ApoA, which is synthesized by the liver, and low density lipoproteins by ApoB, also synthesized by the liver, both of which grab up fatty acids and monoglycerides and cholesterol in the bloodstream so that it's not loose, floating, dissolved in the plasma, but rather connected to proteins. Proteins that either have a lot of extra cholesterol in them, which is very low in density, 0.7 grams per mil compared to water is 1 gram per mil, so low density. And that cholesterol is transported throughout the bloodstream to be available when needed in different tissues, as well as the fatty acids monoglycerides being transported that way. The high density lipoprotein binds more tightly and less cholesterol to ApoA, and it gets taken back into the liver and processed. The extra cholesterol is excreted in the bile. So the high density lipoproteins are protected because they're pulling cholesterol out of the system, out of the bloodstream, and sending it to the bile, which ends up in the intestines and excreted in the feces. So this is a get rid of your cholesterol mechanism. The LDLs tend to release their cholesterol. They're a little bit more loosely bound, binding their cholesterol into the subepithelium, the subendothelial space, and make these atherosclerotic plaques that we talked about before. That's why they're a bit on the dangerous side. Final question. When we fast, and this is going perhaps one slide into the next lecture, because we're going to start talking about catabolism in this slide. We're breaking down things when we're fasting. So this is to get us a start on the idea of catabolism and anabolism, both of which are connected to our dietary cycle. When are we fed? When are we fasting? So here's a preview into catabolism. When we fast, our dietary source of glucose is stopped, right? We quit eating, that's what fasting means. Well, the brain depends on glucose. How does it survive? Go. Okay, G3 wonders if we use glycogen stores, and he's absolutely right, actually. He's righter than everyone else. We first break down glycogen stores for a while, and we have plenty of that available to make glucose. We probably don't do much gluconeogenesis to keep our brain alive. The brain gets the first dibs on the glucose. It's the organ that needs it the most. But it's also true, what all the rest of you said, which is that ketone bodies released during beta oxidation of fatty acids in the tissues, in the mitochondria, can be used by the brain perfectly well to keep the brain alive. So if we fasted a really long time, or we didn't eat any starches, any carbohydrates, no plants, no glycogen for a really long time, we could survive just fine on ketone bodies. That's it for today. Thank you all for coming.